everybody, and welcome to CrownCast, the new podcast from Crown Bioscience that aims to discuss the emerging trends, breakthroughs, and viewpoints in oncology, drug discovery and development. I will be your host for today's podcast. My name is Jonny McMichael, and it's my honour to bring you interesting conversations with scientific experts from around the globe. I'm Crown's Vice President of Client Experience and Enablement. I've been with Crown for coming up for 10 years, and as you can probably tell from my accent, I am based in Scotland, a very cold and beautiful outpost in the north of Great Britain. So for today's episode, I'm very pleased to have with me Dr. Leo Price, who is Crown's Senior Vice President of Corporate Innovation, based in Leiden in the Netherlands. Leo has been with Crown for around 3.5 years, since the merger between Ocello and Crown Bioscience in 2021. Ocello are a leading provider of high-content imaging services for oncology drug discovery, amongst other indications. And Leo has also served for 10 years as the CEO of Ocello, following the founding of the company. So today's topic will be organoids, which is a topic of great interest and love to Leo. He's certainly an expert in this area. So Leo, hearty welcome to you. It's lovely to have you on here. I'm very pleased to spend time with you. So how is Leiden today? Is it almost nighttime there?
Almost nighttime and cold. Thanks very much for inviting me for this podcast, Jonny.
Absolutely, my pleasure, Leo, as always. And as you can tell from Leo's accent, despite being based in the Netherlands and despite what a lot of people may think, Leo was in fact English. Right, Leo? So, Leo, why don't we start off by please tell us a little bit about yourself and how you came to be sitting where you are today.
Yeah, Jonny, so I'm a cell biologist by training. After a PhD in cell physiology, I did a postdoc in California at Scripps in cell biology, and I was working on cell adhesion. And that was a really amazing learning experience. And during that time, I really started to get a feel for using cells and cell culture to study the topics I was working on. And I realized there, looking at cell adhesion, how cells stick to their environment, how they stick to each other, that was where I realized that the current paradigm, the current gold standards of using 2D cell culture really weren't up to the scratch to be able to ask those questions. So if you want to learn how cells interact with each other and what happens in cells after they start attaching to their matrix proteins, if your model basically resembles a fried egg sitting on a piece of plastic, these are not really relevant to the biology that's going on inside the body. And so that was really what made me start thinking differently about the kind of models we should use to study these kind of biological activities. And so that was what really got me into 3D cell culture and organoids, and that's going to be the topic of the conversation. After that postdoc inspirational postdoc in California, I moved to the Netherlands, working in the Dutch Cancer Institute and also in Utrecht, and that was really where I started getting my first exposure to started working with 3D cell culture about 20 odd years ago. And I guess we'll cover some of the questions around organoids and 3D culture in the rest of this talk.
Absolutely, yeah, absolutely, Leo. So I remember when I first heard about organoids a few years ago. I think I was still working as a sales chap with Crown at that time, and I have to admit, back then I knew even less about organoids than I do now, and it was a real step forward for what Crown Bioscience are offering, but certainly what's available out in the drug development field. So before we start talking about organoids, can you tell us a little bit about what organoids are and how they came to prominence?
Well, there's a lot of different interpretations, but I think my take on that is that really organoids are a specialized cell line. There are a couple of key characteristics, and one is that they're driven by stem cell compartment, and those stem cells they then proliferate and they produce progeny which differentiate. And one of the really unique factors about organoids, I think, is that if you look inside a tissue, it's coming from a cultured organoid tissue. It's coming from a single stem cell, but in there you'll see all various different kinds of differentiated cells of the organ that organoid reflects. So I think that that's a key factor. The culture is driven by various different, by very specialized media that drive the stem cells, prevent the differentiation until that's desired. I think another important thing is that these cells never see plastic. Plastic, once those cells will stick down on a plastic, they go through all sorts of changes. Plastic forms a very strong kind of selection pressure, like most in vitro cell culture. And so keeping cells permanently in three-dimensional culture really prevents that extreme selection pressure and keeps the cells more faithfully close to the organ that they came from. So these technologies have been around for really a long time. I mean, back in the 1990s, I think one of the pioneers, probably the pioneer, is Mina Bissell with mammary gland cultures. She started, she was using Matrigel, which is, remarkable material and not yet reproduced synthetically, and she could grow these mammary glands in 3D culture and use those to study behavior, and then also moved into breast cancer as well. And so she was a real pioneer there. She called them 3D mammary gland cultures. But in sort of maybe 2010, in Hans Clevers's lab in Utrecht, I think some really important steps were made there, particularly by Nick Barker, who was a postdoc in his lab, who discovered a stem cell marker, LGR5, and then the ligand, which is R-spondin, and a few other factors which allow stem cells to be maintained in culture. So these are sort of niche factors that from the environment of the tissue that keep the stem cells as stem cells. And so identifying those stem cell markers and those factors really opened the door for developing new methodology to grow normal tissues in culture, which is something that had been really challenging, had just not been possible before then. So that was done in intestinal tissues, and I think that used that breakthrough in being able to grow normal intestinal tissues in a dish gave the confidence to start exploring other kinds of tissues, other kinds of epithelial tissues, and then from there onto disease models, including cancer. So I think that those, and also they coined the term sort of, I think they coined the term organoids, or they started really labeling this technology as organoid technology, and that's stuck ever since. So I think that those discoveries in Utrecht, and then built on by many others, allow the development of methodologies to allow organoids to be grown reliably and consistently, so you can grow these cultured tissues really for years and years, and they just don't change. So I think that those have been really important. Those are important landmarks. Other really important factors that have moved the technology forward is the clinical validation. So showing that an organoid, cancer organoids in culture respond to drugs the same as the patients respond in the clinic. So you can make an organoid from a patient's tumor, you give them the same drugs, and you give the drug in vitro, you give the drug to the patient, and there's a very high correlation between the response in the patient and the response in the dish.
Yep.
And that gives confidence, which cell lines and other in vitro models and mouse models you can't do.
Yeah, so it's that level of translatability that just didn't exist with the existing platforms and systems. I was going to ask you what drove the need for development of organoids. You've sort of already touched upon that, but one of the questions I had for you was, how are organoids used by researchers? Are the organoids used for basic research, for translational studies? How do you see that out in the market now?
I think that the real drive to use organoids was the sort of realization that the cancer cell lines and patients, really there was a huge difference there. As I mentioned earlier, cancer cell lines, they've got a lot of great uses, but they've really diverged a lot from the patient tissue, and we really needed in vitro models that were really brought up closer to the biological relevance of humans. I think another really thing that's sort of lacking in cancer cell lines was the fact that they're all different. This Brent's cancer cell line from this institute, it's cultured for 20 years on plastic in this kind of medium, and another cancer cell line's cultured in that medium. And I think that one of the things about the organoid technology is that they're all, you can collect the tissues and generate organoids and culture them all in exactly the same way. So you have a sort of in vitro patient cohort, and that being able to reflect the patient population is also one of the things that's driven or has been embraced. That's one of the great positive sides of organoids, that you can have those populations. And how they're used by researchers, everything from basic research up to clinical diagnostics, and I think you can look at mechanisms of cell biology, tissue organization, embryology, all sorts of different fundamental basics in any single organ you like, from fat up to brain. I think that these kind of basic research questions can be addressed using organoids, and all the way up to the clinic. So there are now studies going on where you can take a biopsy from a patient, generate an organoid, test them with a drug, and then determine whether that patient should receive that drug or not. And so these are starting to become accepted. So that's really the full spectrum of
almost a companion diagnostic level, almost once you get
to bedside, I guess.
Yeah, exactly. That's perfectly well. Thank you. That's very interesting. And we hear. I mean, I will speak as a layperson in this case. We hear about other technologies like tumoroids, and sometimes it's not clear what differentiates what's different between these different approaches. So how do organoids differ from other in vitro ex vivo technologies such as tumoroids, for example?
Well, again, it's a sort of nomenclature issue here, but I think tumoroids, either way I see that or define that, is basically cell lines that are grown in 3D, and they do recapitulate a lot of biology you would see in an organoid. You can take a 2D cell line, you put it in 3D, and it will then form a organotypic like structure. And so that environment that the cells grow in really has a profound impact on the biology. So tissues will also start to form different cell types, and you form an organotypic structure. But keeping in mind that these have come from a cell line that's drifted enormously from the original patient. So the correlation to a patient outcome is going to be much more unpredictable. The history of that material is also very poorly defined. There are other technical aspects to it. I think the organoids is really a process as well as a sort of a biology, and so the method of culturing is critical there. And I think with tumoroids, it's very anything goes. There are other kinds of 3D. Sorry, Jonny. There are other kinds of 3D biology. From hanging drop, you can go from very simple, moving all the way to using patient tissues, and that's maybe something we can touch on later.
Yeah, absolutely, Leo. Yeah, that clears a few things up for me. Thank you very much. I appreciate that. And one of the last points you made actually segued into the next question I had down here for you, which was, are all organoids equal? Are there significant differences in the protocols and approaches that researchers may use to develop an organoid?
Well, there are certainly different ways of using organoids, and I think the first methodologies were really protocols were well defined by the Clavers group, and these methodologies were then moved into the Hub Foundation, which is was a foundation for utilizing and exploiting these organoids. They're very strictly adhered to, and when I first started working with organoids over 10 years ago with the Ubrecht and with the Hub Foundation, we very rigidly stick to these protocols, and they ensure that the organoid that you start off with is the same one that you're working with in 1 month, 3 months, a year's time. And we know that deviating and cutting corners and not strictly following the protocols and having a very stringent QA, that you will get drift and you will get unexpected deviations in the outcome. So I think that's really the main the main concern for me is to ensure that the protocols that are followed, that we know will preserve the biology that we think we
And
have.
and I guess that becomes particularly important when we move these technologies into the CRO world, for example, where drug development companies, clients, partners expect consistency, expect reliability, and that quality.
Absolutely, Johnny. It's like cell lines. If you get a HepG2 cell and you do a study with HepG2 cells, so liver cancer cell line, from 10 different CROs or 10 different labs, even if you just get them sequenced, you'll find that there
Yeah,
are differences between them, and certainly there will be in terms of the way they're cultured and how drugs will respond. And so it is a real risk. And I think that's also a concern for the users of organoids, is that we do want to make sure that the approved protocols established protocols are being followed, to make sure that we don't have dirty results being generated, and which kind of undermines the confidence in the technology.
that's right. That's right. Because a drug development partner will expect similar results when they come back for the second, third, fourth, fifth project with the same compound, for example. So that consistency is key. Thank you, Leo. That's perfect. It makes perfect sense to me. So are organoids a common part of drug development today? Have you seen that level of adoption?
There's, the adoption is it's gradually increasing. There is a very clear increase in acceptance of organoids, and they're being more and more used in taking more and more of a prominent role in drug development. And it varies between different companies, between different kinds of drug types, where these are being used and embraced. There are, but the first drugs are now coming into the clinic that have been developed using organoids, and there are drugs now in phase 3 clinical trials where essentially all of the in vitro work has been done using organoids. And so I think that other companies will stick to their gold standards for a bit longer, and will be slower to incorporate the organoids. But as more and more drugs develop, the successes continue to be released into the clinic, I think there'll be more and more adoption. And perhaps other drivers will come in as there's more pressure to demonstrate that in vitro methods have been exhausted before in vivo studies are used. So it's just a question of time, I think, before they become really mainstream. But it's getting there.
Yeah, absolutely. I remember in those early days, again, as I was back client facing in those days, and I think we believed at the beginning that pharma companies would be interested in the organoid platform, but actually it was a slow build with pharma, because pharma needed to be convinced of the reproducibility of the adoption out there by regulators, by other pharma companies, and it was the biotechs who were the early adopters, and then that's we've seen a real shift in that balance, I think, in the last sort of 2 to 3 years.
Yeah.
So again, I think you've touched on a couple of these points already. One of the things I was wanting to ask you was what factors you felt were driving the increased use of organoids. You've obviously talked about the translational aspects has been a key part, but for example, do factors such as alternatives to in vivo models play a part in that? Are there other factors you see that drive the use of organoids?
Well, I think that the increased complexity of the targets is certainly going to be a factor. Targeted therapies. Gone are the days where you can take a panel of 10 or 20 breast cancer cell lines and just test a single drug, a chemo, and see whether it works or not. So really, you need to have models which are very specifically represent the indication you're going for and the particular characteristics of the tumor, the particular mutations, and that's something that you can do with organoids. That is that there are hundreds and hundreds of, for example, we have several hundred organoid models which are all sequenced, and if we want to target homologous recombination deficiency, we can pick out those organoids that have the specific mutations and see whether the drug is active in those and how that compares to other organoids or other mutations. But another important factor is being able to compare the effect of a drug on a tumor and on the normal tissue from the same patient. And so this can give you a sort of therapeutic window, can really help with sort of, I guess, the first aspects of safety evaluation. So I think that those are, those are big drivers. I think also other things driving the use of organoids is the increased standardization of the methodology, and increased reliability of the outcome.
Yeah, and publications that are out there talking about
publications. That's right. I mean, the clinical studies. I mean, I think the first ones came out in 2018. Flakogiannis published science paper in gastrointestinal cancer showing really high correlation between in vitro and in the clinic, and there've been 30, 40 or more clinical studies published since then showing the correlation. That's going to drive acceptance. You can't get that with a cell line or a mouse model even.
Absolutely. Okay, perfect. Thank you. Completely hear you. And I was going to ask you again. You have already touched on a couple of these, but if you could talk a little bit on the strengths, weaknesses, challenges with organoids compared to 2D and mouse models, for example.
Well, I think the strengths is easy. There are three main things. It's the biology, and the biology. Those are the three most important strengths of organoids, to labour the pun there. I think the challenges are, as with all new technologies, is a standardization. Another thing has been, I think one of the things that's actually driven the uptake of organoids has been the introduction of methodologies that just work with organoids. So equipment manufacturers have been producing machines that can handle organoids. Reagent producers have been generating the reagents that are required. And basically the whole kind of infrastructure required to work with organoids has been picking up, responding to that demand. And so I think the technical challenges have really, those being overcome, particularly liquid handling and etc. These have really enabled organoids to get mainstream. I mean, when I was first working with organoids, I was pipetting them onto a cover slip and then dipping them in this with tweezers and dipping them in that, and now we're doing them in 384 well plates and using machines, and that technological developments are probably the biggest driver.
The availability of reagents that are applicable, not having to make your own cocktails of reagents.
So it used to be a weakness, but that's less so.
Okay.
Jonny, I think just one point to add. I think, what we want to do with organoids is to have the convenience of a conventional 2D cell line and the biological quality of in vivo. And that, I think, is what organoids more or less managed to achieve. To start with, we didn't have the convenience of the 2D, but I think we're now able to do high throughput screening and screen 100 models in one day and test tens of thousands of compounds. So, it's maybe not high throughput screening as our friends in pharma would describe it, but it's really getting close there. And you're not able to work with those kind of numbers unless you have a really standardized methodology. So,
and I guess within China's the obvious example is that ability to transition the work from the in vitro ex vivo context into the in vivo context, and we have developed PDX derived organoids, for example. We have the ability to take that same genetic characteristics and take them into an in vivo context. So that allows that translation, which again you can't get with cell lines, but not quite to that same extent.
That's right. I mean, translation to the clinic is really important. In the drug discovery process, you still need to have in vivo data to validate your compounds. And so translation to those in vivo models is also really important. And that's one of the things that we've done in Crown is made hundreds and hundreds of organoid models from the PDXs. And there, again, as with the 2D versus patient, we have the organoid in vivo really bringing the level of the biology close to each other to improve the translation between those two systems. And and also to be able to go back from in vivo data or clinical data and go back to the organoids to help you understand what was going on. I mean, quite a surprising proportion of the work that we do with organoids now is not, a prequel to the in vivo models. It's coming back and analyzing, explaining the results that were observed in the clinic or in an animal.
Yeah, it's a great point. And again, it's very powerful to be able to do that and investigate further. So coming to the end, I think, Leo, getting near to the end, what do you see as the future of organoids? And with that, are there other technologies emerging to compete with or complement the use of organoids?
I think that the continued technological development, just improvements on production of organoids is going to be one that's definitely going to be important. It's still a very kind of manual process to produce them. I think that there's going to be leaps and bounds there, probably synthetic substrates to grow them and totally animal-free media to produce organoids. I expect we'll get there. Generating models from patients with drug resistance, the metastatic patients with drug resistance, that's going to be an important one. So really where you see that the organoids that are out there start representing the patient groups that are targeted by therapeutics. So that's the sort of development of the existing technology. But I think other advances need to be in the space of immuno-oncology, despite, some improvements. Really, we're not able to recapitulate the immune system in vitro with organoids. And so, drugs like checkpoint inhibitors don't really work as they do in patients. But hope springs eternal, and I'm sure we'll get there at some point. In terms of one solution there, of course, is not just to try to reconstitute something in a dish, but to go with if it ain't broke, don't fix it. And we can maybe start with tumor tissue, which has an intact stromal compartment. It has immune cells, and actually use those for creating so-called P0 passage zero organoids. And that's something we do in Crown with the ex vivo patient tissue platform. Tissue from the patient within 24 hours, it's going into 384 well plate as a sort of micro organoid-like structures, and there we're able to test immunotherapies, including checkpoint inhibitors. So maybe we'll see that kind of ex vivo technology expanding, being used more commonly. access to tissues is going to be an important one, and having agreements with healthcare providers and patients that facilitate access to those will help move that forward. But the human biology is intact, and if we, the less we change things, the closer the less we change that biology in vitro, the more predictive these models are going to be. So I think we'll see advances there.
Yeah, the future is very bright, and it'll be fascinating to see which directions this field goes in the coming years. And I know there are a lot of innovative companies out there developing various platforms that may overlap with one another, and would be interested to see how even the competitive environment develops over the next few years. So, Leo, do you have any closing remarks or comments to share with our listeners?
I don't know. I think working with organoids and those culture tissues has been really thrilling. From the first experiments that I started doing with organoids maybe 20 years ago, and looking down the microscope. I mean, for cell biologists, doing cell culture is already really exciting and a lot of fun to see cells creeping around. But when you start growing tissues, it's amazing what you see. And certainly when you're putting tissue from patients into culture, you get all sorts of amazing things being created, and you can start seeing kind of cardiac tissue pulsating. I've seen that with my own eyes, and jumped out of my skin when I saw
Absolutely.
this. It's really, as a biologist, it's fascinating biology to work with and really exciting. So, yeah, and so I encourage people to give it a go, if only to stimulate their their enthusiasm for biology.
Absolutely, Leo. Absolutely, and I know what you mean on that excitement. I remember the first day I successfully managed to grow mouse tracheal cells at a near liquid interface and saw them cilia beating, and you're right, it blew my mind. And I guess that's the level of excitement that the scientific research brings, and we should all kind of bear that in mind, right? So it's perfect. So, Leo, as always, it's been an absolute pleasure speaking with you. I really appreciate you taking some time to share your knowledge and views on the world of organoids with us. Hopefully there'll be another opportunity in the future to talk again. So thank you kindly, sir, for your time.
Thank you, Jonny.
And for those listening out there, thank you also for supporting CrownCast. Hopefully you found it enjoyable. If you did, please be sure to hit subscribe on YouTube or Spotify. You can also find CrownCast directly on crownbio.com. So hopefully you may join us again for the next episode of CrownCast, and if so, we look forward to seeing you then. So thank you very much.